My name is Gary Struhl, and I am the Herbert and Florence Irving Professor of Genetics and Development at the Vagelos College of Physicians and Surgeons at Columbia University, and also a member of the Zuckerman Institute. My contribution to this body of work has been to understand what is spatial information, how is it generated, how is it received and interpreted, and how does it change cell behaviors to create patterns. My work on Notch really relates to that last aspect of the problem, which is local communication for cells to coordinate their behavior. So the prize is for basically the discovery of that system, its understanding what its role is, and then ultimately getting to the molecular mechanism by which that system allows cells to communicate. And more than just communicate, cells have to talk to each other, but they also have to listen and change their behavior and talk back so that they coordinate, so they do the right thing relative to each other. For me, it's been very valuable to be paired with Iva because we both have this deep conviction that this is an important way to understand life. And the award recognizes not just my work, it recognizes the approach and the field and the importance of basic research. And for me, I take it as I'm a representative for a much larger enterprise. There are two main reasons why Drosophila fruit flies are my favorite organism for discovery. First, I have a lifelong fascination with using genetics to understand basic biological phenomena. And what fascinates me about genetics is that it allows me to, in effect, reach inside the organism from the outside, to get right inside individual cells and change their working parts, the protein products of individual genes, and then look at what happens to those cells and to their neighbors. And after over 100 years of research in flies, we now have an incredibly powerful repertoire of tools for doing such experiments. And in fact, that repertoire gets better and better every year. And these tools allow us to identify most of the working parts underlying any given biological process you might be interested in, and to delve into both the logic and even the molecular mechanisms by which they work. And the special value of using such genetic approaches is that they tell you what's really happening in vivo, in the living tissues that will construct the animal. So rather than taking the animal and its cells apart to learn how they work, we learn by setting genetic traps that tell us what happens in living cells and tissues in the intact animal. The second reason is that the fundamental question I'm most interested in is the control of growth and patterning. And the incredibly powerful genetic tools we have in Drosophila are exactly what we need to discover how cells communicate with each other to decide what to do. In terms of what challenges did I face when I began to do work in science, and there really weren't any structural challenges. I mean, in other words, it was a welcoming environment for kids and then students who were interested in doing research. I think the challenges are internal. It's your uncertainty about whether you will be able to do it well. And I think what I learned as a graduate student, when I was very unsure about whether I had what it would take to do science, is I realized it didn't matter what I thought about whether or not I'd be able to do it. I should just do it and let the cards fall where they would. And because I couldn't change whether I would have the ability to do it by worrying about it, I should just proceed and let it play out the way it would. And I stopped worrying about that, and that relieved me of a major challenge to my introduction to doing science.